Hello, this is part 3 of 3 in microbiology, the metabolism chapter. And we're looking at how organisms can make ATP without oxygen in a process called fermentation. And what we're looking at is a picture of some beer brewing vessels that are fermenting the sugars in hops and in malt and ingredients that make beer. They ferment the sugars in those substances to make beer. And the one yeast cell that does that is Saccharomyces cerevisiae. Okay, some things to know about fermentation. Fermentation does not require oxygen, and so the whole process is called anaerobic. It converts pyruvate. So remember pyruvate, where did pyruvate come from? It came from glycolysis, right? Oh, here it is. Pyruvate made in glycolysis to an organic product to recycle its electron carrier. So instead of pyruvate moving through to the Krebs cycle and then to the electron transport chain, it stops right at pyruvate and then takes a left turn and it does something else because oxygen needs to be there in order for Krebs and the electron transport chain to go. Because we are creating an organic product as we ferment, these products are usually in 3 classes. They are acids, alcohols, and gases. So acids, you might have heard of lactic acid or acetic acid. Acetic acid is basically vinegar. Lactic acid is acid that's found in like yogurts and things like that to make it sour. Fermentation does make ATP, right? But only a little bit, 1 to 2 ATP per molecule of glucose. Do you remember how many ATPs were made in oxidative phosphorylation? 32 is the low. Let's do 38 because that was our theoretical high. So 38 ATPs versus 2, right? So if you were a microbe, which process would give you the bigger bang for your buck, right? You have 1 sugar molecule. Would you like to have 38 ATPs out of that 1 sugar molecule, or would you like to have 2? Obviously, it would be best to have 38 ATP molecules from that 1 sugar. But some microbes have evolved to live in these very, very low or no oxygen environments, and they ferment to make their ATP because oxygen is not available. Here are some fun fermentation products and the organisms that produce them. So you don't need to know any of them except for our Saccharomyces. You do need to know Saccharomyces cerevisiae. This is our yeast, right? So this is a fungus, right? This is a yeast. All of these are just bacteria, Propionibacteria. But you can see that the similarity of all the different kinds of fermentation that are available start off with glucose and end with pyruvic acid. So this is basically glycolysis, right? Glycolysis happens in the cytoplasm of both of those eukaryotes and prokaryotes. And let's say Propionibacterium is fermenting. When it ferments, it makes a gas and an acid, and it happens to be used in making Swiss cheese. And so what's fun about Swiss cheese is that those holes, right, are the carbon dioxide gas that was produced by the bacteria as it was fermenting the milk sugar, right? So it's trapped in that milk. The bacteria is eating it up, fermenting it, making the acid, which gives Swiss cheese its sort of flavor, and then also making holes in it because the gas buildup creates those little gas bubbles. Lactic acid is one you might recognize actually from yogurt ingredient lists is lactobacillus. So lactobacillus, if you look at your yogurts in the ingredients, you'll find this guy, and it's going to make lactic acid as it ferments the milk products. So yogurt is here as some of that. We can also use it for cheddar cheese and soy sauce. Saccharomyces is going to produce, it does an alcoholic fermentation, so it produces a gas and an alcohol, right? Alcohols are one of the organic products. and we use this to make wine and beer. And the bubbly part of beer and the bubbles in like champagne, that's naturally occurring with CO2. So this is the bubbles. However, in like industry where they actually sort of put the bubbles back in at the end, so it's not sort of like true from the microbe, but if you do it at home, right, if something's fermenting with Saccharomyces, then you'll have CO2 and ethanol being made. And then Clostridium can make acetone and isopropanol. These are two organic byproducts, and then you can actually, like, acetone's basically nail polish remover, and isopropanol is what rubbing alcohol is. So really interesting side products, right? So we have acids, alcohols, and gases. So this is a schematic of fermentation. Again, we have pyruvic acid, right, formed from glycolysis that's going to be converted to carbon dioxide. And then there's an intermediate here called acetaldehyde. Then acetaldehyde gets converted to ethanol. And the reason why we have this step is because we need to recycle our electron carriers. So remember, their job was to gain those electrons, right, from our organic product here. This is our organic, like our starter molecule. We're going to take those electrons. Well, actually this, yeah, I guess NADH is really using acetaldehyde as its organic. So let's just start there. So acetaldehyde is its organic donor. So the acetaldehyde will donate its electrons to NADH. Oh, I'm sorry. I'm kind of messing up here. All right. This is NADH. It's actually giving up its electrons to make ethanol to NAD+. Sorry, the first step isn't shown here, but the key idea, excuse me for my mess up, the key idea is that we need to recycle our electron carriers. So the fermentation process, the reason why we make this organic molecule at the end is because we need something that's going to help us recycle our electron carriers. So in this case, we can see that the NADH is going to lose its electrons to acetaldehyde to make ethanol and then recycle to the empty form of itself. This is another look at a different kind of fermentation. This is called lactic acid fermentation. So same thing here, we have glycolysis, right? Nothing changes for glycolysis. And look at the two ATPs. This is where our ATPs come from when organisms ferment, okay? It's just the two made from glycolysis. And notice, right, we're picking up those electrons. NAD+ is picking up those electrons, but we need to then recycle, right? We need to get rid of those electrons so we can recycle it and take those electrons away. So that's why pyruvate is made into some kind of organic molecule so that we can donate those electrons and then recycle our electron carrier to the empty form. So instead of like going into the Krebs cycle, pyruvate gets converted into lactic acid by accepting those two electrons, and that's why it's called lactic acid fermentation, because we're making lactic acid at the end. Okay, and the lactobacillus, right? Lactobacillus tells you that it makes that lactic acid. Okay, so this is like just a fun idea, like a fun example. Sourdough bread is made with a yeast that ferments the sugars in a starter. So if you've ever made sourdough bread and know anything about sourdough bread, you do have to have a starter mixture, which is yeast and sugars. And so you have to keep this in a sealed container because what you want to do is force the yeast to ferment. So let's review. Remember yeast. Yeast are facultative anaerobes. Okay, so facultative, I can't write the whole thing out. Facultative anaerobes. What that means, and this is what chapter 9 really concentrates on, is learning about the different organisms and their oxygen use. Facultative anaerobes can use oxygen when it's present, and then they can also survive without oxygen and ferment when oxygen is not present. Okay, they ferment. And there are many facultative anaerobes out there, okay? But you would assume, like, if you use oxygen and you get 38 ATPs, This is the preferred method, right? You get so much more ATPs when oxygen's around. So oxygen, if there's oxygen or no oxygen, like it'll definitely use oxygen. It'll definitely undergo cellular respiration if oxygen's present. However, if they're trapped in an environment where the oxygen levels are very low, then they can go ahead and ferment to make their ATP. The products that Saccharomyces cerevisiae here creates is lactic acid that gives you and acetic acid, which gives you the sour taste in sourdough bread. Okay, so sourdough bread, a lot of people are very protective of their mother to make their sourdough. However, if we're just making regular bread, right? If we're making bread at home, we don't use a starter. We don't have that fermentation, right? So we just use Saccharomyces. We still use that same yeast, right? When you get yeast to make bread at home, it's Saccharomyces cerevisiae. It's the same yeast also to make beer and wine. But why is bread not alcoholic, right? Because we know that this yeast does make alcohol with fermentation. And the answer is because when you make bread, you're not in a sealed container, right? You're in your kitchen, you're kneading the dough with all this oxygen around. So Saccharomyces cerevisiae is not going to ferment. It will just use cellular respiration because there's oxygen around to make ATP. So what are the gases, like what are the waste products with cellular respiration? We know that water is a waste product. And isn't there a gas? Right? CO2. So when your bread rises, it's due to the fact that your yeast is eating all of that sugar and, in the bread, the flour is a sugar source, and it makes carbon dioxide gas, and that gas will build up and then rise your bread. Okay, so we're going to do a little comparison here. Cellular respiration, fermentation. So cellular respiration, where is it, right? Where is it? So in prokaryotes and in eukaryotes, cellular respiration is either in the cytoplasm. I have to abbreviate here because I can't write everything very quickly. So cytoplasm for the Krebs cycle and glycolysis. The cytoplasm for eukaryotes is for glycolysis. But then the eukaryotes, since they have mitochondria, they'll move the Krebs cycle and the electron transport chain into the mitochondria. Whereas the prokaryotes are going to have their membrane on the cellular membrane, right? So just that upper cell membrane is where they're going to have the electron transport chain. Fermentation, because fermentation only has glycolysis involved, glycolysis is happening in the cytoplasm for both of our guys. So for bacteria and also for eukaryotic cells, it happens in the cytoplasm. The condition, what I mean here is like oxygen, no oxygen. So oxygen has to be present for cellular respiration to occur, and oxygen is not present, right, for fermentation to occur. What's our final electron acceptor in cellular respiration? That's going to be oxygen, right? I'll just put O2 here. And what's our final electron acceptor if it's in fermentation? It's an organic molecule, right? And remember, this final electron acceptor is the thing that recycles NADH and becomes NAD plus again. So an organic molecule serves as the final electron acceptor. For cellular respiration, what are the products? What do we get out of it? So we get ATP. Let's just go ahead and put the number of ATP here. We'll put the maximum, the theoretical max here, 38. How much does fermentation give you? 2, right? And this is from the same glucose molecule. So cellular respiration is the way to go. It's more efficient. What products are made? So products made are water. This is a waste product, but still a product. CO2, right? And then we have our ATP. What's our products of fermentation? So fermentation is sort of an incomplete breakdown of sugar. So we always get some kind of organic molecule. So I'm just going to org for organic, which can be an acid, or it can be an alcohol. Okay. alcohol. And then as a side sort of product that can be made, not organic, but a gas, right? Sometimes gases are made. So these three are considered the products of, like, possible products of fermentation. Okay, and then we have some sources I've already put in there for you. And some questions to wrap up. The type of fermentation observed in yeast is? Alcoholic fermentation. Glucose molecule during the process of glycolysis is broken down into, hopefully this is easy, 2 molecules of pyruvic acid. And then what is the primary role of NAD+ in fermentation? And you should recognize that it acts as an electron acceptor. And that's it.